All right. So we're gonna go and review the lophotrochozoans, or sorry, lophotrochozoans. So the two groups you need to know are the annelids, which are the segmented worms, and the mollusks, which are the gastropods, the snails, cephalopods, the octopi and the squid, bivalves, clams, and the chitons, which are the polyplacophores. So we'll get into those a little bit later. Just to backtrack though, the lecture that has the lophotrochozoans, where we talk about the two groups of protostomes. Protostomes. I know this is already in a previous lecture or a previous YouTube video, but remember, so it's a pretty poorly drawn tree of life, but at this base right here, these are all the bilateria. So all these groups are bilaterally symmetric, and you divide those into the protostomes, which we'll talk about today, which are the annelids and the mollusks, and then you have the deuterosomes. Um, the differences between these two guys are all just based on, as far as you need to know, based on their developmental strategies. So in the protostomes, so there's three things you need to know: how they differ in terms of symmetry, of division, the determination of 8-cell embryo, and blastopore fate. All right. So real quickly, so this is the checklist of three things you need to memorize, differentiate between protostomes and deuterosomes. I wouldn't worry too much about anything else besides these things. Just remember these three things, okay? So number one, symmetry. The protostomes, what they do, what is it called? Spiral cleavage. Whereas the deuterosomes do radial cleavage. And what that means is at that, so you start out with a fertilized egg, right? So this is an early embryo, it's just one cell, divides into two cells, divides into four cells. Once it's four cells, it wants to divide upwards to 8 cells, right? To make the 8-cell embryo. In spiral cleavage, when it divides to 8 cells, each one of these cells divides upwards, but they'll divide when they split on like a kind of shifted uh, axis. So the resulting 8-cell embryo looks, I don't know if I can draw this well. Let's try that again. Looks like, ooh, that's ugly. Not bad. Okay, so you can see at the bottom right here, these are the original 4 cells, and these are the 4 new cells. But the cleavage between the split cells in the second layer and the first layer are offset, and that's what makes spiral cleavage. And so when they divide, they keep on dividing upwards like that in a rotation kind of fashion. Whereas the radial cleavage organisms at the 8-cell embryo, they will look like this. So these are the 4 cells at the top, and these are the 4 cells below. And you can see the cells above, the cleavage lines are directly over the cleavage lines of the 4 cells below. So that's what makes radial cleavage. So in the protostomes, they have spiral cleavage. In the deuterosomes, they have radial cleavage. Next, in each of the 8 cells in the deuterosome, if you take one of these cells away and pop it off and put it in a petri dish, it'll grow into a new embryo because each one of these 8 cells is cleaving, is differentiating indeterminately, which means that there's no specific fate for those cells at this time in the development of the embryo. In the spirally cleaved protostome, each one of these cells already has a specific fate. So if you remove one, the resulting animal that grows out of that will be missing a large chunk of its tissue. Right? Each one of these cells already has a specific fate designated for it in the future. Cool. Finally, so after 8 cells, we'll go into 16, 32, so on and so forth. Eventually, it'll form this hollow ball of cells called a blastula. And past this, the walls of the blastula will invaginate. They'll go inwards to form this kind of bowl-like structure. And the opening of that punched-in section, that invaginated section right here, is called the blastopore. In protostomes, this blastopore becomes the mouth. In the deuterostomes, this becomes the anus. And that's just something you got to memorize. Okay? Easy. So, backtracking through the whole tree of life of animals, you have the sponges right here, you have the cnidarians and the ctenophores right here. Then now you have the bilateria over here. This is what we're looking at, and specifically today, or in this specific lecture, we're looking at the protostomes right here. Protostomes, the deuterostomes over here, or down here. And within the protostomes, we're going to look specifically at, so the protostome, we highlight this one section right here. It'll fork into 2 groups that you need to know. Lophotrochozoans. I don't know if I spelled that right. And the excidozoans. So we're going to look at these specifically today. Again, these are the segmented worms, the annelids, and the mollusks. The difference between these two, again, as far as you need to know, is that the excidozoans, they shed their exoskeletons. Okay, so let's go on to the specifics of these groups. Number 1, the annelids. These are the segmented worms, and the main animal that is representative of these guys that you need to know that we went over in class are the earthworms. So there is another kind of worm that you're responsible for that is the nematodes. So they both look like worms, but the way you differentiate them is that the annelids, they have segments. Pretty easy to tell, right? So some quick biology about the annelids. Number 1, they're truly coelomate. So all these bilateria, all the protostomes, all the deuterostomes that you need to know are all truly, they have a true coelom except the nematodes, which are in the excidozoans. So we'll talk about that in the next video, but don't worry about that. So that's the exception to the rule. Everything else is truly coelomate. Okay. A couple of quick biological tips about these guys. Again, they have true coeloms. They have these segments. Each one of these segments is a metamer. So the whole idea of segmentation is called metamerization. And one of the reasons of having these segments is that each segment is kind of sectioned off from the rest of the body, and it has its own group of organs, and allows for some amount of functional redundancy. So in each segment of the earthworm, if you'll recall, so each segment, except the specialized front and the back segments, it'll have kidneys, the nephridia, it'll have locomotory body parts. So the setae, the hairs on the sides, and in maybe other body shapes of annelids, they also have these parapods, which are these fleshy outward growths of their body, which they both function as locomotory organs, so they help move the annelid through its environment. So if you cut off one segment of this annelid, you can still filter your blood with the kidneys and you can still move around. So it allows you to stay alive and move and filter your blood even if you've lost some of your body. Okay, so that's redundancy. So that's one of the reasons why having metamers and having metamerism or segmentation is good. Another thing, besides the parapods, so setae, these hairs, setae, and then parapods below, both used for dragging the worm through some sediment or whatever. It also has a hydrostatic skeleton, and we've talked about this a bunch of times. I won't really go into details of what that means. You should know by now. Just know that you should know the groups that require hydrostatic skeletons. So those would be the cnidarians, the nematodes, the annelids, and the echinoderms. Okay. And finally, they're monoecious. Means most of the worms are hermaphroditic, so they have both male and female sex organs on the same animal. Cool. The next thing that we went over, there's a dissected earthworm, as you remember. I'm not sure how much detail you need to know about the dissected earthworm, but I'm going to go over basically just all the parts that we looked at. Okay. And so I'll just I'll define and name the parts and give you their purpose, and then I'm just going to leave it at that. You can decide how you want to study that personally. Uh, once this video is out, once Monday comes, I can no longer kind of direct your studying. So this is my wild guess. I'm just going to shotgun everything at you, and then you can deal with the fallout. Right. So you have the prostomium, which is the front. That basically is the part of the front segment, which is a specialized uh, piece of the earthworm that is right above the mouth. So you can think of that as the part of the earthworm which is associated with the mouth. There's the mouth opening right here. The pharynx goes down this way, right? It's where all the food goes down. You've got hearts. I think there's 5 hearts in the front of the earthworm. Then you have these big white glands, the calciferous glands. The calciferous glands, they collect and then release calcium. You have the seminal receptacle, which holds semen, and then you have the seminal vesicles, which make semen. So seminal vesicles make and hold semen that the earthworm itself makes, and the seminal receptacle is the female organ which takes in semen from another earthworm and holds it in. Then you have the crop, which holds food. Then you have the gizzard, which grinds up food. You'll have septa in the middle, which are the internal manifestations of the segments. They just are connective tissue that kind of segment out each different segment of the earthworm internally. Then you have the intestine, where you start digesting stuff. You have a dorsal blood vessel right here. There's also 2 ventral blood vessels, I think, on the top of the earthworm. Again, I'm just going over the slides here. It's important for you to go into your lab notebook and fill in the gaps that are not discussed in these audio notes. Okay. Then you have the typhosole, which is another digestive organ. And then that's pretty much it for this slide. Other things you need to know, again, the prostomium, which is in the front, the clitellum, which is that large segment of the earthworm, right? Every earthworm has a kind of a larger segment. That is That is where the cocoon with the eggs is held around the worm itself, and where eventually the young come out of. Okay, so it's a reproductive organ. You have the pygidium, which is the kind of the opposite of the prostomium. Pygidium, the structure associated with the anus. Then you have the setae, which are the hairs on each segment that drag the worm through the ground. The earthworms themselves don't have very prominent protopods, or sorry, parapods. So those are in more marine annelids, for example. They have, in addition to setae, they have these large, kind of fleshy extensions of their hydrostatic skeleton that act as fake feet. And that's the earthworm. Now we're going to move on to mollusks. Also, remember in the earthworm, sorry, real quick, you have longitudinal muscles and circular muscles. Longitudinal muscles shorten the worm, and circular muscles lengthen the worm. When you put the action of these two muscle groups together, you get this pulsing of the worm through the media itself. So it kind of pulses the worm forward, right? A piece of the worm will kind of contract and lengthen, and it'll push forward the worm. That ripple will go through the worm, and it'll push it forward. And that's called peristaltic movement. Okay. All right, mollusks. So mollusks, some unifying features. It's important to like kind of get an example of because they're so diverse in how they look. Let's take a look at the generalized mollusk. So for the generalized mollusk, you have a digestive tract, right? Food goes in here, goes through here, and comes out the anus. So this is a cross section of that mollusk. You have on top of the mollusk a shell. The ancestor of all mollusks had shells. Some of them still have shells, some of them don't. The ones that don't either have a secondary kind of shell that got really, really reduced that's now stuck inside the mollusk, or it's lost it completely. But the ancestor of all mollusks had a shell. So below that shell is the mantle. The mantle kind of covers the fleshy covering of the main body of the organism that has all the internal organs, right? So this is the mantle. Most mollusks have a big fleshy lobe on the bottom, which is called the foot, or the head foot. And then often they have feeding structures right here. It can be a radula or a beak, or those are the two feeding organs associated with the mouth. Radula, beak, depending on the group. And then within the mantle, remember the mantle is the main body mass, there is an opening that is open to the environment that has this leafy thing right here. We'll draw that in a different color. This leafy thing, that didn't work. Right here. That leafy thing is called the cnidium. And that's the gill of the mollusk. Okay, so these are the shared features between all mollusks. This is a generalized mollusk right here. Now I'm going to go into the different classes that you need to know. Number one, the gastropod. Those are your snails. Pretty obvious what a snail looks like. You have your mouth. The mouth always or usually has well-defined radula. And a radula is a kind of a bony tongue. These can be turned into different things, but in most of these snails, they have a bony tongue that's kind of a rasping organ that like kind of scrapes algae off of rock surfaces. Okay, so you've got a radula, you got a fleshy foot, you have a shell. The GI tract kind of curves upwards back towards the top of the shell. So this is where the anus is. the anus actually opens up into the mantle cavity. Remember, the mantle cavity is this cavity, this opening within the mantle that houses the tenidia. It'll always house tenidia. So this is where the tenidia is inside the snail. The one thing about the gastropods that kind of deviate from this general body structure are the slugs. I don't think there were any land-dwelling slugs in your lab, but there were a lot of sea slugs, and they can be maybe difficult to tell if you. So superficially, they might kind of look like worms, but they're a lot flatter and a lot more irregular looking. They kind of flatter looking animal like that. Versus the two worms that you're required to know, one of them has segments, the other one doesn't, but they are both really, really thin. They look like worms. So this looks more like a flatworm maybe, but you don't have to know any flatworms. So it'll be pretty distinct. So this is still a gastropod. It's just a gastropod that's lost its shell. Some of these sea slugs or nudibranchs will have the tenidia as a leafy organ in the back. Others have them internally, but regardless, they don't have shells. Most of them are either very prettily colored, like brightly colored to warn predators about their toxins, or they're clear. So those are the two groups that you saw in lab. They're either clear or very beautifully colored. Okay. So those are still gastropods. Just remember that. It's kind of an exception. The only thing that might look like a gastropod, so the gastropod, I think the main focus on that is the head foot, or the stomach foot, the gastropod. That's what it means, right? That's that big fleshy foot organ that is the stomach of the snail that moves it around. There's another group of so everything that has that fleshy foot that sticks to the ground is going to be a gastropod, except one group that you also saw in lab, which are the polyplacophores, which are the chitons. So they look like this. So they have this fleshy foot. Their tenidia are actually in a skirt around the bottom, but you might not be able to see it, but you'll see the foot for sure. The way to differentiate these guys foolproof from the gastropods is that they have these plaques, these multiple shells on top of the chitin. That protects the chitin, but it gives it flexibility to allow it to roll into a ball if it wants to float away. Some identifying features, they'll often have eye spots on the shells, which can identify, simple eyes can identify light and dark and maybe coarse vision. I'm not exactly sure, but they're definitely good for seeing light and dark. They have a radula in their mouth because they're primarily algae eaters, and they have a thick foot, like we talked about with the gastropods. This thick foot anchors them to and pastes them to the surface on which they're eating, so they're hard to remove. Okay, just remember anything with a foot is going to be a gastropod, except the ones with multiple shells, which will be a chitin. Polyplacophores. Again, remember the scientific names. Next, we'll have the bivalves. You can look more deeply into the very complicated structure of how they are organized in your lab notebook. What I'm going to say that you should be aware of are they have two shells, and their tenidia. So their tenidia are arranged on these big leafy gills that are on both sides of the fleshy foot in the mantle. So it's this basically a clam is basically so this is a snail with a shell like that. Clam is basically a snail that fell on its side like this, and that one shell turned into two shells. So that's a clam. So the tenidia will be on both sides of the main body of the clam itself, the foot and the mantle. And these large tenidia are also called lamellae. And they act as not just as respiratory organs, but they also capture food because all these clams and all these bivalves are filter feeders. So their mouth organ or their very specific organs inside the main mass of the body will come and take the food from these gills and transfer them to an opening which counts as the mouth of the clam. Other things to know, they've got abductor muscles which help open and close the shell. They might have siphons to direct the water more efficiently to their lamellae, and they can swim and move around, sometimes either using their fleshy foot, which is in the middle, which they can just kind of like poke outside of their shell and like move it around and dig, or they can flap the two sides of their shells and swim, kind of like scallops do. Okay. Finally, we'll go into the squid and the octopi and the nautiluses and the cuttlefish. These are the cephalopods, and they're very distinct. A lot of the classic mollusk features are just unrecognizable in these guys. They'll have tentacles with suckers on them to capture prey. They have, in addition to a radula, they also have a beak in the middle of all these tentacles to chomp down on food. They're all predatory. Uh, they'll have a siphon right here, which is connected to mantle cavity with the tenidia, which opens up into the mouth and the anus. The mouth and the anus come. Here's the whole digestive tract. It opens up into the mantle cavity, which is also containing the tenidia. And this large mantle cavity can pump water through it and through the siphon outwards to act as jet propulsion. So the octopi specifically can move around with their tentacles that crawl around the floor, but they can also jet water out their siphon. The squid primarily rely on water coming out of their siphon to uh, to move. They'll also The octopi have no shells. The squid and the cuttlefish still have either a small or relatively large shell inside of the mantle. They also have well-developed eyes. Yeah. Finally, just like before, just want to go over the kind of basics of the dissection of the squid that you went through. So you'll have this large gonad up here. It's either a testes or an ovary. If it's a testes, if they're testes, then there'll be a penis that comes out of the side, and there will be a large gland that makes the spermatophores. If it's a female, you'll have a nematema gland, which deals with egg production, but regardless, there'll be a gonad, and there will be an associated gland next to it or below it. Next, we have the ink sac. There's this like silver ink sac in the middle. These cephalopods will spray ink in order to get away from predators, so they have ink sacs. There will be two tenidia on each side, these finger-like projections, and attached to these tenidia will be two sets of hearts. One you'll have, let's see, so you have paired hearts over here, which connect to a central heart down here. This is the siphon side, so there'll be a siphon right here, and so these tenidia are housed in the chamber, the mantle cavity, which then opens up into the siphon. On the other side, you'll have the pen, which is the very thin reduction of the original shell that is like connected, is embedded inside the mantle covering, which gives it some structure to move the water as a torpedo. And then you'll have the optic nerve and stuff like that in the front of the squid. Won't go into too much detail about that. You've got the eyes, and you'll have the mouth, which has a beak in between all these tentacles. And I think that pretty much covers the annelids and the mollusks. So these are all the lophotrochozoans.